Okay, thank you, Adolfo, for your presentation, and thank you for inviting us for present our group for the other member of the table. LS is a complex disease. I think the last year in the other Congress, several speakers, Adriano, to speak about the complexity of disease. But it's true that ELS is a progressive degeneration of after or over motor neuron. Patients die between 2 and 5 years after diagnosis. Several genes have been described for the familial or genetic form, but majority of cases is sporadic. The pathology is very, very complex too. There are several pathways, cells, tissues affected for the disease. And we are lost, confused for this disease. But the reality is we have two big problems in LS. The first is we don't have a therapy, and good therapy, and we don't have the good biomarkers. Our group is the objective treat this big problem because we have the translational research, but we work in veterinary faculty. Then we work with the animal model. The classical animal model for LS is a transgenic animal overexpressed the SOD1 human muted gene. We work with the culture cell and sometimes with the patient too. We work every time the same way. For therapeutic strategy, we perform the preclinical studies. After we patent, and we look for a company. This is not easy. For the biomarkers, it's similar, more or less, but the difference is we study in collaboration with the hospital to study in the patients. Even if we don't know the etiology of disease, we can improve the motor neuron survival. We try this, or even muscular function. For this proposal, in our lab, we have 3 different strategies. The first is based on fragment C of tetanus toxin. Fragment C tetanus toxin retains the retrograde and transsynaptic transport across central nervous system. After intramuscular injection of naked DNA coded for TTC in the model animal, we saw that reaches the protein reaches the spinal cord, increase survival in the animals, and improve motor coordination in the model. Increase the number of motor neurons and decrease reactive microglia. This work was in collaboration with Xavi Navarro. A molecular pathway, TTC, restore apoptotic and survival pathway in animal model. To improve the neuroprotective effect of TTC, we try to make 2 fusion with the other neurotrophic factor, BDNF and GDNF. But the preclinical trial did not show a synergistic effect. With all this work, we have 3 patents, and we licensed for 3 different biotech. The problem was not to attract the interest of big pharma. We thought that we can search other applications for this TTC molecule. Then we study spinal muscular atrophy and ischemia. Spinal muscular atrophy, we have stopped the study for 2 reasons. The first, we don't have good animal model to disease, and now there are better therapies to correct the gene in the clinical trials. Ischemia, the results were fantastic, but the problem was that the injury was made after the therapy with TTC. Now, we are working to apply TTC therapy after ischemia injury. The second strategy is cellular therapy in ALS. Why? Because we have done a PhD and we monstered that muscle satellite cell activity is dysregulated in skeletal muscle for animal model, even in pre-symptomatic stage. The question was, intramuscular transplantation of stem cell in animal model could be therapeutic, and what kind of stem cell should be used? Finally, we decided for the bone marrow cell because we considered that it was easier to arrive to the clinical trials. Bone marrow cell transplantation in the animal extend the lifespan and increase the neurotrophic factor. Maybe this is the effect, and this work was done with the collaboration with Salvador Martinez, and now he started clinical trials using this strategy. And finally, today we speak about this before. I think Merce. Drug repurposing in ALS. Okay, why? Another time. Okay, the cost to introduce a new therapy in the market is high. And after economical crisis, the pharma wants use and drug effective for a disease for other disease. Then, in the same way, to stimulate muscle regeneration, we have used the Neulasta or granulocyte factor estimulante de la colonia, granulocyte colony stimulating factor, GSF, okay? This is commercialized and before perform the clinical trial, wow, the several papers described that this factor promotes mobilization, proliferation, and differentiation of hematopoietic stem cells. And then it stimulates neuronal differentiation or adult neuronal stem cell in the brain. The question was, what's happening in muscle? Because has been described too that hematopoietic cell can help to the skeletal muscle. Before performing the preclinical trial, we study the effect for this factor in proliferation satellite cell, and we observed the proliferation from higher. After intraperitoneal administration for this factor, we can see that increase the survival in the animals and the product reduce the muscle denervation. Now they are in clinical trial with this product. But in our case, we have another question. The therapeutic effect for this factor could be also a consequence of bone marrow mobilization. If that was the case, reducing the hematopoietic stem cell pool should be opposite effect. Antineoplastic 5U were used. And surprise, because the animal live more. Okay, after our surprise, we are a little happy because we have a therapy. The problem now is why? Why the 5FU have this effect? Until now, We know that 5 of the product modulated different blood cells and cellularity in both marrow, but no modulate, it don't modulate motor neuron survival, reactive gliosis, muscle function, and muscle pathway. We research maybe their effect in protein aggregation, neuroinflammation, on microRNA. Help if you want some idea is welcome. The other problem, no good markers. We work every time the same way. We take the different samples in the animal model, and after the variation of expression, number of cells, whatever, is correlated with the longevity in the animals. After, we translate the result to the patient. In the first time with Hospital 12 de Octubre, we work close with Alberto García Redondo y Jesús Esteban, and after with other hospital, national and international hospital. This is the current biomarkers we have studied in the lab. The first is mRNA and muscle and blood. We started this study in 2005, and we start with the studying the skeletal muscle. We compare the rate in presymptomatic animals, and we observed that 72 genes were deregulated. After, the most dysregulated genes were tested along the disease. And after, with the biopsy, we observed only 5 genes correlated with the longevity of the animal. And this is translation for the patient. This is the study. There is a multi-center study. We have 3 different LS patients, other neurodegenerative patients, and controls, and 2 kinds of samples, blood and muscle. The main result obtained was that in skeletal muscle, collagen 19, gene expression and protein expression was a stronger biomarker to diagnosis level. You can see here, this is the patient for the other control. In blood, not only collagen 19, if the diagnosis, MIF2C and sortinexin, this mRNA gene expression, were also identified as biomarkers to support diagnosis. Prognosis level. Low level of collagen 19 were associated with a slower progression of the disease. The other kind of biomarker was microRNA in plasma. After the study in the muscle, we have selected several. We studied 2 muscle different, fast fever and slow fever. Fast fever are more affected in the disease, and there are more genes deregulated. Until now, we can say that microRNA 206 is a potential circulating biomarker. Why potential? Because in the animal model, the results are good, but the preliminary results, we have only preliminary results in the patients. You can see here 12 patients and 12 controls. The other, hematopoietic stem cell, a monocyte in blood. Why study this? Because in our opinion, in animal model, there are an impairment in the stem cells. And we want to know if this impairment can use for biomarkers. We stay very happy because some hematopoietic stem cells are correlated with the longevity in the animals. But the problem was that these hematopoietic stem cells were not detected in ALS patients. Then regarding monocytes is different. This is a big correlation too in the animals, and non-inflammatory monocytes show a positive correlation. The animal has more non-inflammatory monocytes live more, and for the inflammatory monocytes were contrary. Now we're waiting for the results in the patient in collaboration in the first time with Hospital 12 de Octubre. Cytokine in plasma, because every group described the cytokine in patient, animal models. Okay, after a lot of work, a lot of money, 1 PhD, this is fun, this is good, and frustration, circulating cytokine, our conclusion is circulating cytokine could not be good prognostic biomarker in mouse model of ALS. We don't waiting for nothing because we make a little essay, a little study in patients, and we didn't know the significant difference. And now we belong to the European Trial Consortium. The website will launch the last November. This consortium, several countries are together to arrive to increase the clinical trial, because for like this, several countries can increase the number of patients, and we are there. This is the team. Anna, Chris, and Jane are here. They speak much better English than me, but it's my role today to speak. And close collaboration, Hospital 12 de Octubre, Xavi Navarro, the other people, punctual collaborations in the international and national. And now we are very happy in this fever, and we want to collaborate, if you want, with the sample, with the product, with the model, and this is the most important collaboration with us. Thank you very much for your attention.
Thank you so much for your perfect adjustment to the scheduled time. We have time for a couple of questions. Any question? Rafael?
So I have a question. So what is the mechanism that you think is behind the protective effect of this tetanus toxin in terms of molecular mechanism? Because, you know, the effect of tetanus toxin is well known, the target is known, it's an air protein. So what do you think is behind that?
Okay. When we start with this work, the idea was use fragment of tetanus toxin like a right to the central nervous system neurotrophic factor. This is the idea, because we have, this is studying Pasteur Institute. For Pasteur Institute was important like neuronal transfer, but we try to a ver que me sale en francés. To reach the central nervous system with the neurotrophic factor. But after, we can see that not only fragment C of tetanus toxin. We didn't know the receptor, but Dr. Aguilera in Barcelona, he showed that TTC can use the different receptor for neurotrophic factor. This is normal because it's normal because normally I'm not born for infected for tetanus toxin. But we didn't know very well.
Any more? No, thank you, Rosario.